about that. But thanks all for having me, and it's fun to be in this exciting session. And I feel bad for having this long 15-minute talk because there were so many fantastic lightning talks. Well, this is actually a preliminary idea that I'm going through, so bear with me. I think lots of us in the field, especially since the advent of speciation genomics, have been really interested in trying to understand what might influence patterns of introgression across the genome. I think this is this great obsession that the field is dealing with right now. And there's been good reason for, so you can see these sorts of plots where you can see across the genome, you calculate DXY or FST or something and tell some stories about what's going on. And I think there's very good reason for this. We want to know what's different between species and how they maintain their distinction upon secondary contact. Okay, and the things that people are often interested in are these canonical intrinsic incompatibilities, these Jansky-Muller incompatibilities, where you have, one allele substitute in one background, another in the next. They've never seen each other before, and they can be bad. There's also a lot of interest in, you have two organisms adapted, to different environments. They come together, and their offspring could be poorly adapted to both environments. But I think that there's maybe more simple things that could be at play. They could prevent introgression, and I want to work through those. And I think that the ways that organisms reproduce, how they transit their gametes from one generation to the next in the genetic environment that they are in, might have a major role in influencing patterns of introgression. And I think that selfers and outcrossers might highlight some of these differences. And so you probably know this because you're in this session, but selfing plants pollinate themselves. Outcrossing, and so their mom is dad. Outcrossing plants can get pollen from anyone. Okay? And there's differences that come across with those lifestyles. All right, so we think that selfing plants have small effective population sizes, small population recombination rates. This can lead to an increase in the number of mildly deleterious mutations. But because they show their worst sides every generation, they purge their recessive lethals or bad recessive. Okay, outcrossers sort of the opposite. They can do a great job of getting rid of mildly deleterious mutations, but perhaps less good at these recessive alleles that are often hidden in the diploid state. Okay, and so I want to know if this can explain what we see when we have two plants of different mating systems in the field and we start to see hybridization between them. It seems like we see hybridization and introgression between selfing and outcrossing plants quite often. Here are two examples that I think are starting to establish a broader pattern that you often see. Here's Mimulus Nasudis indutatus with work by Amanda Kenny out of Andreas Weigart's lab. And you can see here this yellow part of the structure plot corresponds to individuals that are morphologically Nasudis. And then as you go here, this is the outcrossing guys, and you can see that all Nasudis guys are largely only sulfur, have only Nasudis ancestry, while Gutatus, the outcrosser, has some sort of quantitative variation from some to little ancestry from sulfur. Okay, you see a similar thing here. This is Dave Muller's work in Clarkia. Again, the X's are flipped. Here's the sulfur. It has almost no ancestry from the outcrosser. Here's the outcrosser. It has some amount of ancestry from the sulfur. Okay, so how far can this basic sort of simple difference in genetics get us to understanding what we see in these crosses, okay, or what we see in the world? And again, there's two parts to this. The idea is that the mildly deleterious alleles that can accumulate in sulfurs will be removed by more efficient selection when they head into this higher NE lifestyle of the outcrossers, so that should prevent introgression in one way. In the opposite direction, these rare deleterious recessives that everyone has should be barred from homozygosing and should not make it into a sulfur. Okay, and so mildly deleterious strands actually didn't do any direct work here because there's recently been really nice work out of Graham Proop's lab by Ivan Jurek, and also similarly really good work by Kelly Harris and Rasmus Nielsen, where they've looked at the case of human and Neanderthals where Neanderthals are meant to have smaller effective population sizes than humans. And here's a figure out of Ivan's paper, where he's found that as there's higher gene density, there's less Neanderthal ancestry in humans, suggesting that mildly deleterious mutations can disfavor ancestry from a population with a small effective population size in a larger effective population size species. Here's some theory from Kelly Harris that kind of does similar things. You can see that if you have like less ancestry from Neanderthals here, when you're near mildly deleterious recessive, that can accumulate Neanderthals but not humans. So that same sort of thing should be going on in plant and selfing outcrossing situations. Obviously, although Neanderthals have small effective population sizes, they can't self. And so we have to think about this other problem for the recessive lethals when we think about this other direction. So this sort of thing has been appreciated before, that you take a selfer and an outcrosser, you make an F1, you self them, and what you can see is that, you know, the guys that have inherited two recessive lethals at a given locus will not do well. And so that should push against bringing outcrossing ancestry into the selfer. This was pointed out actually as a technical artifact in a nice paper on genome by Gresham and collaborators, where they said, oh, we went to the field, we did some new hybrid stuff, and we found no F2, simple F2s, or self-F2s, and they thought, well, maybe that's just because you can't interpret them because you're missing some of these markers because you're selecting against outcrossers. Okay, so what can we do? Here's some simulations. What I've done here is, here's very selfing, here's very outcrossing, and I have a I'm, some centimorgans away from a recessive lethal, and you can see the probability that a neutral allele will fix. Okay, and so when you're highly selfing, a neutral allele will have a really low probability of fixing. I scaled this to make it a percentage and multiply it by 2 times the number of chromosomes in the population, so that up here would mean that it didn't matter. Okay, and so that's what you get. Okay, so it seems like this can act. Exposing recessive lethals can prevent, or at least try to push against introgression outcrossing ancestry into selfers. When you look at this plot, now what I'm doing is I'm thinking about unlinked recessives, and I'm plotting here as a function of the outcrossing rate. Again, what's the probability of fixation? And if you can see these numbers, don't listen to them or don't trust them. This is the case if I have 1 recessive lethal with some outcrossing rate, if I have 2 recessive lethals, 3 or 4 on average per genome, and it turns out that the probability of that neutral allele unlinked to any recessive lethals will have fixation probability of 1 minus 1/2 raised to the number of recessive lethals in that genome, so it's got to survive there, and you can multiply that by the selfing rate. So you can pretty simply see that even if you're unlinked to a recessive lethal, that can still select against outcrossing ancestry. Okay, so trying to put it all together, both parts of this thing make sense. I've tried to figure out how I can, if they can work together. And so I've coded up some forward simulations where I isolate 2 populations for some number of generations. I then have secondary contact. I just thought about 1 chromosome that's a Morgan long. And I've played with the number of loci that can get hit, the frequency of recessive lethals, and the mutation rates, population sizes, migration rates, etc. And I just want to show you 2 extremes of what's going on. And the problem is I haven't found too much in between these extremes, so I'm trying to think about where I should go with it. So here's a forward simulation. I've had outcrossers have a population size of 5,000, selfers 500, and this is a sort of case where the outcrossing ancestry is favored in both selfers and outcrossers. Okay, so even though these 2 processes going on, in this case, there are so many mildly deleterious mutations. So here in blue is the proportion of selfing ancestry, and under our selective simulation in gray under neutrality. So you can see here all these bad mildly deleterious mutations are kept out of the outcrossing ancestry, but in fact, the selfer will just trade out some of those recessive lethals for being better. that sort of pattern under some parts of parameter space. Under other parts of parameter space, we get the exact opposite result. Here, there's so many recessive lethals in the outcrosser, it would rather have some pretty terrible mildly deleterious mutations and move them around. Okay, so we get both answers, and I've yet to, I haven't done anything yet particularly systematic, but we're yet to find a case where these two things balance out, and that you get more, more selfing ancestry than you should in sulfurs, and less selfing ancestry than you should in outcrossers. So that's roughly where I am, and I have some directions that I could go in, and I would love to chat to you about them. One is I had a fixed fitness effect. I could pick from a more realistic distribution of fitness effects. I could think about this across divergence time as we move on the speciation continuum between sulfurs and outcrossers. These basic patterns might change. I could think about being more realistic in terms of heterogeneity and the recombination rate and the density of bad mutations across the genome, because these are realistic things that happen in the world. Or I could just scrap this. And so, let me know what you think, because I can't tell what I do. Okay, so that's where we're at. I'd like to thank some people for their comments, and you, because you'll hopefully give me other great comments and you came to listen. All right, thanks a lot. Any questions? Yeah.
Yeah, so you showed us the two extremes, and you were saying you hadn't really found anything between. I was wondering, which parameters do you think you'd need to modify in your simulation to get something more in between?
Probably a lot of them. and so I'm having so it's really hard, at least for me, to develop realistic loads by simulation. You have this problem where sometimes, maybe I guess Jeremiah how great he did, but sometimes you just all crash and the sulfurs get really bad. Because if you have an NES like such that bad mutations will fix in the outcrossers and not in the sulfurs, then you can just get like an infinite number of them piling up, or as many as you have sites. And so it feels a little bit artificial. It's part of my concern about how to do a good job with this. So I could play with sort of the number of recessive lethals, the number of mildly deleterious mutations, and the selective coefficients. But I've been struggling a little bit to think about what's a fair and reasonable way to do this. So if anyone has any good ideas, I'd love to hear them. Well, thanks Jeremiah for the session.